Can you hear me, please?
Yes, doctor. Please start your presentation.
Okay. Hello, everybody. My name is Nuhun Traore. I'm from Burkina Faso, and from the Institut de Recherche en Sciences de la Santé. So I'm really, it is a really pleasure for me to be at this Congress to present you the synthesis of my work on the genetic diversity and population structure of the invasive Oriental fruit fly Bactrocera dorsalis in Burkina Faso. So, in introduction, we can say that mango production is about 300,000 tons per year in Burkina Faso. This is about 56% of all fruit production. The mango is also the major export product and also a raw material for industries. And it is also source of income in a rural area and a source of food security. Unfortunately, one of the main problems of mango production is the infestation by Bactrocera dorsalis indel. Bactrocera dorsalis indel in its life cycle, after mating, the female lays eggs in the host fruit pulp, and the eggs develop to larvae, and these larvae in their development destroy the mango quality. Is this complex contain about 100 species. 100 species we have set including Bactrocera dorsalis indel, Bactrocera Bactrocera candiensis, Bactrocera folia, and trivialis. In this group, the most problematic species are Bactrocera dorsalis indel and Bactrocera carambole. Bactrocera dorsalis is a pest of major economic importance. It is a
Hello, doctor. No one. Are you there? Doctor, no one. Please respond.
Yes. Yes, I'm here.
Yes, doctor. Please share your screen again.
Okay, okay. You want me to come back or I can continue?
Yes, doctor, I can able to see now. Please continue your presentation.
Is it okay?
Yes.
Okay, thank you. Bactrocera dorsalis is a pest of economic importance, worldwide distributed, and it was detected in Africa uh, in 2003, detected in Burkina Faso in So, excuse me, I think that I have internet issues. So I sent you the record of the presentation. Maybe you can present this if
Yes,
possible.
doctor, we have your recorded video. I'll play it. Please just give me 2 minutes.
Yeah, yeah. Okay. I send you since 13 minutes. Institut de recherche en sciences de la santé. It's a real pleasure for me to be at this fourth conference on this fourth conference on sustainable agriculture and food system. So I will present you the synthesis of my work on the genetic diversity and population structure of the invasive Oriental fruit fly, Bactrocera dorsalis in Burkina Faso. My presentation will follow the plan of introduction, material method, research, discussion, and conclusion. In introduction, we can say that mango production is about 300,000 tons per year in Burkina Faso, and this is 56% of all fruit production. Mango is also a major export product, and it is also raw material for industries. Mango is also a source of income and a source of food security. Unfortunately, the mango production face the infestation of Bactrocera dorsalis. Bactrocera dorsalis in its life cycle the female lay after mating, the female lays eggs in the fruit host pulp, and these eggs will develop to larvae, and the development of these larvae contribute to the degradation of fruit quality. Bactrocera dorsalis is a fruit fly, part of the complex Bactrocera dorsalis CL. In this complex, we have around 100 species, and between these 100 species, we have 7 horticultural pests, including Bactrocera dorsalis indel, Bactrocera carambola, Bactrocera candiensis, Carie occipitalis, pirifolie, and trivialis. But the most problematic species are Bactrocera dorsalis indel and Bactrocera carambola. Bactrocera dorsalis is a pest of economic importance. Its distribution is worldwide. It was detected in Africa in 2003, and in Burkina Faso in 2005. But nowadays, it is the main pest of mango production, and contributing to sometimes at 100% of losses in the mango orchard. The current control method against Bactrocera dorsalis are based on the sanitation measure, the chemical control, and also the biological control, and also the IPM and the genetic control in prospect. The prerequisites for Bactrocera dorsalis or any other pest control is to understand consists to understand the ecology, the biology, and also the population genetics. That is why the aim of our study was to investigate the population genetics of Bactrocera dorsalis in western part of Burkina Faso. For that, we first investigated the genetic diversity, and secondly, we investigated the genetic structure. In the part material of method, we collected our examples in western part of Burkina Faso, and the example concern the male individual Bactrocera dorsalis, and in male Bactrocera dorsalis collected in seven localities, and in each localities we collected in 3 plant formation, including mango orchard, Chi agroforestry park, and natural formation. And then after the collection, after the sampling, we analyzed the individuals by morphological identification using the key morphological identification for Bactrocera dorsalis, including a dark thorax with 2 yellow costal bands, and also a form of a mark of T form in the or on the abdomen. And then we counted the individuals by type of plant formation and by locality. And then we did our statistical analysis on our software. And then we sampled 315 individuals for the population genetic analysis. And between these 315 individuals, we collected 45 individuals per locality. So these samples were analyzed by PCR using 10 microsatellite markers. And after PCR, we did electrophoresis analysis to observe the different migration band. So the microsatellite marker polymorphism by Ben or was undertaken by Ben analysis, and the information concerning the genotype of every individual was collected in a database, and this database was analyzed then to undertake the genetic diversity and genetic structure using R software and GenAlex software. The result show that the maximum density of flies were observed in Kenedougou with 2,986 flies per day. The minimum density were observed in Sicily with 87 flies per trap per day. And these distributions were statistically significant according to ANOVA test. In fact, Kenedougou constitutes the main food production area in Burkina Faso, according to Weroro et al. Bougriba, Pomoe, Oué, Kenedougou, and Pony, where we observe high densities are located in southern Sudan climatic zones, and also Bulkiemde Sangier and Sicily, where we observe low densities are located in Sudano-Sahelian climatic zones. According to Nyangosu et al., Bactrocera dorsalis abundance is strongly correlated with rainfall. Results also show that in plant formations in each localities, the distributions were statistically significant according to P-square tests. And we observed that the high densities in each localities were observed in mango orchard. In fact, mango constitute the preferred host fruit of Bactrocera dorsalis in Burkina Faso, according to Zida and collaborator. And also, mango availability is always correlated with the population growth of Bactrocera dorsalis, according to Mwatawala et al. The results show also that the genetic diversity of Bactrocera dorsalis by localities was high, with an average of 0.722. And also the fixation indexes were positive in all localities, with an average of 0.166. This means that we have a deviation from Adiwember equilibrium with heterozygote deficiency. The results also show that the gene flow between all localities, were high, with a maximum of 27.53 between Pony and Pomoe, and a minimum of 10.62 between Kenedu and Sicily. The results also show that the genetic differentiation between all localities were very low. The genetic differentiations between all localities were very low, with a maximum of 0.023 between Pony and Pomoe, and also a minimum of 0.009 between Sicily and Kenedu. In fact, these results observed are due to the high dispersal ability of Bactrocera dorsalis, and also the fruit exchange, the density of fruit exchange between localities. The result also show that the genetic structuring in Bactrocera dorsalis by Bayesian admixture analysis. The results also show that the Bayesian admixture analysis show no evidence of geographical structuring. However, it showed that we have 3 ancestral genetic groups in Burkina Faso. The result also show that with discriminant analysis of a principal component, the result also show that by the discriminant analysis of the principal component, we have 3 weakly differentiated clusters in Burkina Faso. And between these three clusters, we have Oue, Pomoe, Buguriba, Pony, Sicily in the first one, and also the individuals from Kenedu in the second one, and the individuals from Bulkemde, Sangue in the third one. In fact, Kenedu, being a major area of fruit production in Burkina Faso, is a kind of enclave with unidirectional flows. It means that we have more genes going out and less gene coming in. And also, Bulkemde Sangue is a kind of little isolated from the other localities by distance. In conclusion, we can say that Bactrocera dorsalis is differentially distributed in Burkina Faso according to locality type and according also to the type of plant formation in each localities. Bactrocera dorsalis population have high genetic variability in Burkina Faso. There are 3 ancestral genetic groups within the population, and we observe no evidence of genetic structuring. However, Kenedu and Bulkemde Sangue appear to be very low. Appear to be very low as isolated from the others. This result will serve to guide the different intervention against Bactrocera dorsalis. Thank you for your attention.